We saw the etylation process when putting the seeds between wet paper towels in a dark area to start growing. Since phytochromes aren't receiving any light, they remain in the PR state, leading to an excess of PR in the plant. Because there is no PFR present to continue the process of germination, the plant became desperate for light and let out skinny white roots in hopes of finding some. We see de-etylation when the seed is transferred from the paper towels to the grower. From here, we see the seeds start to green and branch out. This is due to the phytochromes receiving light, specifically red light, causing it to transform from PR to PFR. From this point onwards, we see an excess of PFR, an active form of phytochrome which allows the plant to start greening. With hydrotropism, we see the roots of the plants mostly extend down into the water deposit of the grower, without not many roots straying out in different directions. With geotropism, we see throughout the first days of the plant that the roots start to grow directly downwards. This is known as positive gravitropism. The shoot the shoot of the plant, on the other hand, grew directly upwards in respect to gravity. This is known as negative gravitropism. With thigmotropism, we saw the plant alter its shape due to its strange environment in the grower, with the roots becoming really flat, for example. With phototropism, we see that the shoot of the plant bends towards the window as it senses a source of light coming through it. To start off with the hormones, we have auxin, which is involved in a lot of growth factors. Firstly, auxin is involved with the elongation of roots. We observe this with the young developing shoot it produced. Next, it is involved with the extensive root system that the plant had formed. Finally, we see that auxin is what caused both geotropism and phototropism to happen in our plant. The changing levels of this hormone cause the plant to bend and form into what it is today. The next hormone, known as cytokinin, is also involved in the growth of the plant. This hormone is what regulates cell division, which we saw when watching the plant grow over a period of time. It also controls accipital dominance, which is where both auxin and cytokinin control growth of the plant. With cytokinin alone, it promotes the growth of the shoots, which we also saw with our plant when it started to green. Expansin, this is our next hormone known as expansin, which is also highly involved in growth. This hormone is located in the cell wall of plants. It is involved by loosening the cell wall, allowing the plant to grow. We saw this in our plant when watching it grow throughout the period of time. Our next hormone, gibberellin, is also involved with the growth of the plant. This hormone plays a factor in stem elongation, which we saw when the plant started to receive light and green. It is also involved in the beginning stages of the seed. This hormone activates seeds to germinate when water is sensed, which we saw after letting the seed germinate in the paper towels. Our next hormone, acetic acid, is seen in the very first stage of this project. Acetic acid was also the reason why our seed isn't growing when we was the reason why we didn't see it grow when we first received it. It kept the internal dwellings of the plant encased until we placed it in the right conditions to start growing. Our last hormone, ephylin, will be part of the beginning stages of the plant. This hormone is what inhibits leaves from sprouting when the shoot is digging to the surface to find light. We saw this in the germination stage, where in the paper towels, the seed let out a shoot to find a light source without branching. Now, if the plant shown grew tall but did not show much branching when growing, we could look into the idea of shade avoidance for an explanation. This phenomenon occurs when a plant is grown in a shaded area. It is due to phytochromes of more elevated plants absorbing most of the red light from the sunlight, leaving rays of far red light to pass through. This plant below is only receiving far red light in its phytochromes, and the subunit known as PR becomes dominant. Since PR is dominant in the plant, the stems begin to grow more while the branching effect becomes more ignored and is receiving little to no growth. The overall objective of this stem elongation is to outcompete the taller plants in height so it can finally receive red light. In order to avoid this kind of growth, it is recommended that you move the plant into direct sunlight where it will become more stubby and bushy due to its easy access of red light. The circadian rhythm that the plant we grew is the circadian rhythm of the plant that we grew is probably one of its most important factors. Plants use this to keep track of the day as well as environmental changes. Without this process, plants would not be maximizing the amount of energy they can consume and the growth that they could have. The plant uses its phytochromes to do this, sensing all changes of wavelengths throughout the day. It is how plants keep track of the day and night cycle, and it can tell like what season we are currently in. In our plant, we witness the circadian rhythm in action while watching this time lapse. This plant would typically sense the light in the morning and instantly start its light-dependent processes until about midday. Then the plant uses the rest of the day to convert the products of these processes into chains of energy. Once dusk falls, the plant uses the entirety of the night to use these chains of energy to stimulate growth, therefore making the plant larger. Without the circadian rhythm in our plant, it would have not shown as much growth as presented in this video.